Hi, everybody. How are you this morning?
Good.
Good. How are you?
Good. That's great. You're healthy and getting through this period in pandemic. So hopefully we'll have a vaccine soon, right? Or maybe even a couple of those. So all we can do is just to stay safe and wait for that to happen. So it's Monday, November 16th, Monday, November 16th. And we are in the middle of chapter 11. So I don't know if we're gonna finish it today or not. Hopefully we will. If not, we will also spend some time on Wednesday talking about it. Now the homework for chapter 11 is available, was available yesterday already, and or even Saturday, I can't remember. And it's gonna be due on Monday of the Thanksgiving week. So you have a week to work on it. So let me share my slides. Share my slides. So we finished with this slide last time by talking about how hydropathy plots can predict transmembrane domains. And we move on to
Dr. Kornienko.
Yeah.
Yeah, on that past slide. So why didn't you count the one right after before the first one? Why didn't you count this first one on part B? So see how you have A. Why isn't that one A?
This one?
Yeah.
And this one is gonna be one, two, three, 7. Yeah, that should be A.
Okay. Okay.
That should be A. I just basically, I don't think I was really paying attention. I just wanted to indicate that the segments which are below 0 in the hydropathy index are exposed to aqueous environment.
I see. Okay.
And the ones which are above are transmembrane domains.
All right. Thank you.
So in general, so we talked about different, how the structure of fatty acids can change melting points, right? Can change melting points and can change how fluid the cell membranes are. And so we know that the cell membranes will be fluid if they have shorter fatty acids, right? So remember, it's all about intermolecular interaction, right? And so the shorter the fatty acid, the weaker the intermolecular interaction and the more fluid the membranes are. And unsaturated, more unsaturated fatty acids because of these kinks that are introduced into the structure, right? So zigzag like this, and then like so, and then things will change, right? So and then if you have a more predominant saturated fatty acid, there will be a gap. There will be a gap, which you can fill in, for example, with cholesterol, right? So we know cholesterol can fill these gaps and make membranes less fluid. All right, so At high temperatures, so depending on the temperature also, so this is not so much from eukaryotes, but many prokaryotes, they live under different conditions at different temperatures, right? There are thermophiles, there are organisms which live at lower temperatures. And so they can and then you start wondering how do they adjust their membrane fluidity, because membrane fluidity is crucial, right? They have to be able to pass certain molecules through, they have to accommodate proteins in the cell membrane, and those have to be fluid to move from one part of cell membrane to another. And so the organisms can do that by changing the type of fatty acid. All right. So, and another way of doing that, so eukaryotic organisms have cholesterol, right? So different types of sterols. Now, fungi also use a molecule that looks like cholesterol, but it's not quite. It's known as ergosterol, right? So there's some structural differences here. And aerobic prokaryotes contain hopanols, so these kind of structures. Also, a multi-ring system with a whole bunch of hydroxy groups and something looks like a sugar on the end. So this will be a polar residue, polar head group. So a number of ways these cells can change the membrane fluidity, and that obviously will affect how fast these lipids can move in a lateral direction, right? So, and they can actually, under normal conditions, so even though we kind of look at these cell membranes as static, thinking that, oh, this lipid is stuck there for a long time. No, not at all. In fact, these lipids can move across, basically making their way through other lipids as fast as 1 micrometer per second. And we have even to put this number into a better perspective. So in E. coli cell, for example, a lipid can circumnavigate the entire cell in 1 second, right? So in other words, basically think about it. So you have a lipid starting on 1, if this is the cell, right? If this is the lipid, it can move around like this. This will be the cell membrane. It's a horrible picture, but it'll give you an idea that this lipid will move around in 1 second. It's amazing. It's amazing how in 1 second, right? So it just tells you how fluid cell membranes are. There's constant motion. So these lipids just move one past another very quickly. Now, there are techniques for us to actually, how do we know that, for example, right? There are techniques for us to measure the rate of lipid diffusion. And so one of them is fluorescence recovery after photobleaching, FRAP. So this is just an overall picture, but let's look at how we do it step by step. So what we're gonna do is so this is our cell, right? So this is the lipid membrane. And we're gonna apply a fluorescent probe or fluorescent dye to label the lipids. So the dye is not going to be penetrable. It will not be able to get into the cell membrane, but it will stick to the polar head group. So the red spheres here is the dye. So so we're going to totally cover, right, the cell with this dye, with fluorescent probe. And if we view this with fluorescent microscope, you can see that the entire surface is covered with this dye, red dye. Then in step 2, we're gonna apply intense laser, right? So this intense laser will excite the dye molecules and basically cause bleaching. So the molecules will not be able to accept any more light, right? And they will be sort of bleached out, and there will be no color coming from this area. And that's the second step, photo bleaching. And then the third one, so we stop the laser and we start observing. We stop the laser, we start observing, and you can see that the dyes molecules of the probe, which were attached to the lipid, they will be slowly diffusing back into this bleached area. Okay, and we can measure the rate of this fluorescence return. Measure the rate of fluorescence return. And again, this is based on the lateral diffusion of the lipids. And depending on how fast this happens, we can tell how fast the lateral diffusion is in the cell membrane. So technique known as FRAP, pretty straightforward. Pretty straightforward. Now there is a different kind of diffusion we also might imagine. So this one is the, you may wonder if there's lipid, let's say on the outside of the cell membrane, if it can undergo diffusion and sort of flip to the cytoplasmic side of the cell membrane. And you can already see this is very slow, right? If we just talked about 1 second of circumnavigation in E. coli for lateral diffusion, for transverse diffusion it's very slow. Why do you think that is? Why is transverse diffusion so slow compared with lateral diffusion?
The polar head has to travel through the hydrophobic region, so it's really unfavorable.
Yep, makes sense. It's very logical, right? So you have a polar head group. How do you drag this polar head group across the lipid bilayer? Not easily. And so you can see this is half-life, right? T1 half, this means half-life. It's measured in days, right? So it requires several days for half of these lipids to undergo this transverse diffusion. Okay, so this will be for spontaneous flip. But we also talked about membrane asymmetry, right? We talked about membrane asymmetry, and we know that the type of lipids in the outer layer are not the same as the types of lipids on the inner layer, and that is maintained. That is maintained by the cell, and there are special enzymes which actually accomplish that kind of transverse migration or flip. So generally, you can call these enzymes by the name flipase, but they're actually different types of flipase, and they actually each have their own name. So the flipase by itself is the molecule which uses the energy of ATP, molecule that uses the energy of ATP, and it will move the, for example, this one, phosphatidyl ethanolamine, or PS, phosphatidylserine, from the outer to cytosolic leaflet. So as you remember, for example, phosphatidylserine predominantly lives in the inner layer of the cell membrane. Do you remember what happens if you have a if you have phosphatidylserine on the outside?
It signals for like apoptosis of phagocytes to come destroy it.
Yeah, that's right. So there is an enzyme that is making sure that no phosphatidylserine shows up spontaneously, or if it does, it removes it. It moves it back into the inner layer, so that this cell by mistake does not get eaten up by the phagocytes. All right, and it uses the energy of ATP, right? So ATP is hydrolyzed in the process to ADP and PI. Now there's FLOPase, which does the opposite. Now we'll talk about, so ATPase and ABC transporters, they're slightly different in their structure, but they both work in the cell membrane. And so this ABC transporter, known as FLOPase, does the opposite, right? So basically it grabs a lipid from the cytosolic leaflet and puts it into the extracellular environment. Now, and there's a third one, there's scramblase. So these enzymes do not really do anything specifically. They don't actually utilize any energy. All they do is they utilize the energy of concentration gradient. So in other words, if you have more of the lipids of the same kind on the inside, it will use this concentration gradient to move some of them on the outside, and vice versa, right? So they don't really move lipids in one particular direction. They just basically lead to equilibrium. They just allow for the lipids to flip, so that there will be 50-50 distribution of the lipids at the end. All right, so commonly these are known as flipases, and very important enzymes for asymmetry. All right, are there any questions?
Are they always transmembrane, the enzymes in the cell membrane?
The enzymes themselves, if they're transmembrane, well, they kind of have to be, right? So they have to operate they have to operate on both sides of the cell membrane, right?
Okay, thanks.
So they have to be transmembrane. Yeah, I know they're drawn as transmembrane, but they also have to be transmembrane. All right, so the membrane is also asymmetric, not only in terms of the kind of lipids on the outer and inner layer, but there's also also non-random or uniform distribution of lipids laterally. Okay, so there is something known as lipid rafts. For a long time, this was actually a very, very hot area of research. And specifically, so these are clusters of sphingolipids with longer than usual tails. So you can see, so the lipid raft will be here. Lipid raft will be here. So you can see the blue is glycerophospholipid, right? And then the red one will be glycosphingolipid. And it's kind of extending slightly, making the membrane slightly thicker at this portion, right, where the sphingolipid is predominant, and then it goes back to the glycerophospholipid. And so so these are, and obviously because and they also stick to each other. You can see there's a lot of cholesterol here. There's a lot of cholesterol, and cholesterol kind of glues these all together. And so lipid rafts, they are lipids which are glued together and they travel all together. And so that's why they're called kind of rafts. So, so the rest of the cell membrane is known as a sea, and this is a raft. in the sea. And actually rafts are not just little rafts in the ocean. They actually account for a very large proportion of cell membrane. Oftentimes the 50% of the entire cell membrane is actually glycosphingolipid rafts. So these are, so you can see these fatty acids here, the red ones, you can see they're longer than usual. They're longer than usual. This is longer than usual. So they're cholesterol rich. They also rich in GPI linked proteins. So we talked about that. Some proteins in lipid rafts, they're doubly acylated proteins, right? So basically they're kind of covalently attached to the cell membrane through double acylation. I think this is primarily on the cysteine residues. On cysteine residues, this is prenylated protein. We talked about farnesyl lipid last time. And so lipid rafts have a variety of functions. Some of the functions of lipid rafts is actually to bring proteins together so the proteins can interact, right? So the proteins can interact, and it's a kind of a way for the cell membrane to not allow for spontaneous random distribution of proteins in cell membrane. In fact, they'll bring these proteins together and make these proteins participate in protein-protein interactions. So they allow segregation of proteins in cell membrane. Any questions? So these are rafts. Okay. So membranes also undergo fusion. So many organelles can fuse with each other. Many different vesicles can use with cell membrane, for example. And they can do that without exposure of lipids to the aqueous content. And whatever is inside can be delivered from inside of one cell membrane to another. So you can see here some examples. So vesicles from Golgi complex. So you know the Golgi is responsible for post-covalent modification of various proteins, for example. And you can see that these proteins then will be packaged into vesicles, and these will be, so it's going to be secretory granule, right? And this will fuse with the cell membrane and allow for this chemically modified protein to leave the cell through exocytosis. Remember, exo is outside, going outside, endo is coming inside. So you can see here something is coming from the outside through, and the cell membrane will allow this fusion. And so many times, for example, this could be a lysosome. You know, one of the main functions of lysosome in human cells is digestion of unwanted proteins, for example. Lysosome. So that's how viruses get inside the cell too, right? Through membrane, from the membrane fusion of sperm and egg. Same idea. Vacuoles in plants. and separation of two plasma membranes and cell division. So, so this fusion can be spontaneous or it can be protein mediated. And there are specific types of proteins which participate in this process. They're known as SNARE. Now there is a longer term for this, but we don't need to remember that. I can't remember. I don't know it's even in a textbook, but I'm sure you can go to Wikipedia. But that's irrelevant as far as what we need to learn about these SNARE proteins. So there are, you can see, okay, let's look. I think we have a, yeah, bigger picture of this. So let's say we have a neurotransmitter filled vesicle. So remember in neural signal transduction, right? So the neurotransmitter, which is present in vesicles. So if this is, let's say, synapse, right? And there is a presynaptic neuron, and there is a postsynaptic neuron. And so inside of this, there will be vesicles with neurotransmitter present. And when the action potential arrives, action potential, action potential. So these, like so. So these neurotransmitter filled vesicles will fuse with the membrane of the presynaptic neuron. So postsynaptic will still be there. And these will have to fuse. I don't know how to draw this well. Into the synapse. So here is the secretory vesicle. This is the vesicle that contains this is the cytosol right here, right? Right here, secretory vesicle, which contains neurotransmitter. So it approaches the plasma membrane. Now there are, there are V-snare vesicle bound proteins, and there's T-snare, it stands for target membrane snare proteins. And there's also another one, there's 3 of them. And there's another snare known as SNAP25 on the plasma membrane. And so the way this works is that all these 3 come together and start making this triple helix. And eventually it will continue spinning around. All 3 will form this tight triple helix, which will start opening the cell membrane, start fusing the 2 membranes together. So for example, the lipids in this area from the two membranes, they give you one. They give you one. And so first, obviously, we're going to fuse the one of the leaflets from each. You can see here, one of the leaflets has been fused. So there's one continuous sort of membrane, right? But the neurotransmitter is still there. So this is known as hemifusion. Hemifusion when outer leaflets of both membranes come into contact. But then the neurotransmitter will still has to get out. And then so these snare proteins continue pulling things away from each other and create an opening, and a neurotransmitter is released. Okay, so so this is protein mediated. This fusion is protein mediated, and there are three snare proteins playing a role. All right, any questions? Okay. All right, so another major topic, transport across membranes. So membranes are they don't only serve a defensive role and don't only separate the cells from each other. They they basically have to allow for especially polar molecules to pass through, and or ions, for example, right? And so there are many different ways to accomplish that. So cell membranes are permeable to small non-polar molecules that passively diffuse. So passive diffusion of polar molecules involves desolvation. So what that means is that if you have a polar molecule, in an aqueous medium, it will be hydrated. Right? And so it will never be able to cross the cell membrane because of all the water attached to it. And so for it to cross the cell membrane, it kind of has to shake off all these water molecules, shake them off, and only then it can cross the cell membrane and then regain the hydration sphere on the other side. So because of that, it has very high activation barrier. So that's regarding passive diffusion, right? So when we don't do anything, just allow for the molecule to diffuse. So it's going to be very slow. But the transport can be facilitated by proteins, for example, that provide an alternative diffusion path, right? So so some transport is passive, but a lot of transport is known as a facilitated transport. And such proteins are called transporters or permeases. So must be energetically favorable. Now you can see here, regardless whether it's passive or facilitated, it has to be energetically favorable. And you can see, for example, this energy can be provided by the concentration gradient, right? So for example, if you have a solute C, and let's say this is a membrane, right, in the middle, you can see there is a high concentration of C1 on one side, a low concentration of C on the other side. And so before equilibrium is reached, there will be net flux. You can see this large blue arrow, right? So most of this solute will be going left to right. If there is a way to cross the cell membrane, again, it can be passive, can be facilitated. But if there is one present, it will be crossing the cell membrane. And eventually it will continue doing so until the concentrations are equal on both sides. So at equilibrium, there'll be no net flux. Now for ions, for example, this will be electrochemical dependence, right? So again, we want to make sure that the potential, electrical potential is similar on both sides, right? Which basically means the concentration of negatively charged particles has to equal the concentration of positively charged particles. And that's not the case here. You can see there are many more positively charged particles on this side and negatively charged particles on that side. And we allow this to equilibrate until eventually the numbers of negative pluses and minuses on the left equals the numbers of pluses and minuses on the right. And so, so when we talk about facilitated, right, diffusion, so transporter proteins, so the problem is, so let's say this is your solute, right, the compound that needs to cross the cell membrane. You can see what's happening. Basically, it's surrounded by this hydration sphere, right? So these are little molecules of water, and molecules of water are two polar, right? So they will obviously hate the lipophilic medium of the cell membrane. So the solute does not have the power to bring all that water across along with it. So this solute has to shake off these molecules of water, become kind of naked. And then once it's naked, then it can enter the cell, if it's lipophilic enough by itself, can cross the cell membrane, get on the other side, and regain the hydration sphere. So this is often referred to as hydration sphere. So, and you can see here, because of that, the free energy is very high, right? So basically, again, we're dealing with, this has to climb all the way up because all those molecules of water have to come off. Then there is slight stabilization because this gets into the lipophilic bilayer and presumably interacts with it positively. And then it loses the interaction with the lipophilic bilayer. And then the energy drops down once it regains the hydration sphere. So the process can be facilitated. And you can see now we see the blue path. Blue path, we have a transporter shown here. And even though with transporter, again, we'll lose the hydration sphere, but hopefully this transporter can compensate for all the energy lost due to the loss of the hydration sphere. So in other words, your solute will bind very strongly to the transporter protein, right? So let's say you have lots of amino acids there, lots of carbonyl groups, lots of NH bonds, right? Hydrogen bonders, hydrogen acceptors, and that will grab the molecule and drag it, or the molecule will go by itself because of the concentration gradient, and will come out on the other side and regain the hydration sphere. So, so this is simple diffusion, and this is facilitated diffusion. So let's then summarize what kind of types of transport we have. So let's start with simple diffusion, just what we had just now. So this should be non-polar compounds only, right? Because they do have to interact with the lipid bilayer when they cross the cell membrane, and it has to be down the concentration gradient, right? So in other words, the concentration of S has to be higher on the outside than it is on the inside. Now, facilitated diffusion, again, down the electrochemical gradient. Again, the concentration of S on the outside has to be higher than on the inside, But this time the molecule doesn't really need to be totally nonpolar, right? And because there is a transporter, that can grab it, help it lose the hydration sphere. It will facilitate, provide lots of hydrogen bonding for it, and allow it to come out on the other side down the concentration gradient. Then there are types of active transport. So the active transport, so compare it with just facilitated diffusion or passive transport. Active transport basically will be achieved against the electrochemical gradient or concentration gradient, depending on if it's a charged species or neutral. And so you can see what's happening here. Now the concentration of the species on the outside is actually smaller than on the inside, but it'll still come in. It'll still come in because there's so this primary transporter will have ATP associated with it, and it will hydrolyze ATP to provide energy to draw the molecule against the concentration gradient. And now there's another one. There's a secondary active. There is secondary active transport against electrochemical gradient, often driven by ions moving down its gradient. So this is kind of coupled. So you can see here, even though again we have a concentration of S is smaller on the outside than on the inside, so it will be moving against the concentration gradient. This transport is coupled with the ion transport, and for the ion, the concentration of ion is actually lower inside than it is outside. So ion is allowed to come in, and it will be coupled for the S that will come in, right? So it's driven by the ion moving down the concentration gradient. Then ions by themselves, right? So here, if it's not primary, it's just a facilitated. Now ions by themselves, again, they can't really cross the cell membrane. So it has to be facilitated transport. So it has to be an ion channel, a protein known as ion channel. And it will allow for the ion to move down the concentration gradient, right, from the outside to inside. And the last type of transport is ionophore mediated ion transport, and that usually happens down the electrochemical gradient. Basically, what this is this is a like an empty shell, right? Empty shell, like so. Empty. So this is a molecule that can grab an ion like so, Now there's this ion. Then it will cross the cell membrane. Cross the cell membrane. And release the release the empty ionophore. And then ionophore then can actually go back, be ready to bring more ions inside the cell. Now, this is a very common mechanism for various prokaryotes. So prokaryotes, which are looking for particularly metal ions in the extracellular media, right? So basically they produce these molecules known as ionophores, which go outside and hunt, basically hunt for ions, grab an ion, bring them in and cross, release inside the cell membrane in a cytosol. And then we'll go for another hunt. So very neat system. All right, so remember this word ionophore. Questions? No questions. So another way to categorize these transport mechanisms. Now it's all logical, all makes sense, right? So there's no really difficult material here. So we're kind of going through these slides pretty quickly. The molecule can, so this is facilitated transport. So the molecule can just go straight through down the concentration gradient, and this will be uniport. Just uniport, moving in one direction. Now, if the two molecules coming in at the same time, in other words, one molecule cannot come in without the other one. So we just looked at this one, like secondary active transport, right? Ion coming in together with the substrate, with the molecule. If that happens, it's a symport. They come in together at the same time, but they don't have to come in the same direction. So the molecule can come in inside when another molecule is coupled to that transport and going outside, let's say down its concentration gradient. So that's known as antiport. So uniport, symport, and antiport. And these two are co-transport. Co-transport because the molecules will move only in the presence of the other molecule. So these are three cases of transport systems. There is a glucose. Shall we do the glucose? Well, we are kind of out of time. It's 9:49, and we'll need more time on glucose. So let's end here. Do you have any questions about today's lecture? Anybody? We're almost at the end of this, so there'll be some discussion on Wednesday. I haven't heard you guys for a while. Can you still hear me?
Yes,
Yeah.
sir.
Oh, thank you. Because you've been quiet for a long time. I'm like, do I still have the audience? All right, so just no questions for today?
No, sir.
All right, so the homework is open. Let me know if you do have questions, write something in discussion, utilize the discussion, and I'll talk to you on Wednesday.
Thank you.
Thank
Bye.
you.
Thank you.
Mhm.